My name is Antonio Somarakis from Leiden University Medical Center, and today I'm going to walk you through ImaCytE, an integrated tool for the visual exploration of cellular microenvironments for imaging mass cytometry data. The motivation for this project stems from the fact that the cells behave differently according to the microenvironment. Hence, our goal is to provide an interactive tool to explore and analyze the cellular microenvironments, as derived from a novel imaging modality, imaging mass cytometry, which provides a stack of more than 40 grayscale images at subcellular resolution, where each image illustrates the abundance of a specific protein in the tissue. The combination of these proteins define the phenotypes of the cells. To get the cells, firstly, the images need to be segmented in a pre-processing step. Input to our workflow are these segmented images. The workflow consists of 3 tasks and starts with inspecting the quality of the samples, interactively identifying cohesive phenotypical groups, and stratifying the phenotypical groups into subgroups with unique microenvironment characteristics. We start with the validation of the quality of our samples, which is focused on the discovery of possible batch effects in order to enable the user to discover such spatial patterns according to the prior knowledge. We used color to encode the expression value of each cell, and the tissue samples are displayed in a small multiples view. Finishing with the quality control, which is the first task of our workflow, we move forward to the second one, which is the identification of the existing phenotypes in the sample. In order to define the different phenotypes, we are using an interactive visual approach presented in our previous work, Cytoexplore. In Cytoexplore, first we are using a dimensionality reduction technique, ATSNE, to visualize the data, and then we are trying to visually identify the clusters in the embedding. Afterwards, using MISShift algorithm, we capture on the fly the visual clusters, or we let the user load a predefined clustering. In order to illustrate the high-dimensional signatures of each cluster, we are using a heat map, where each column represents a cluster as derived from the plot above, and each row the protein. The color represents the median expression value of the given protein in each cluster. In addition to our previous work in Cytoexplore, now we know the exact location of each cell, which plays a critical role for the expert in order to verify the identified phenotypes. Hence, we semantically color code the phenotypes according to their similarities in the high-dimensional space, enabling the user to select one or multiple cells or clusters in any of the 3 views, highlighting the cells in all the others. Having identified the phenotypes, the experts start the exploration of the cellular microenvironment, divided into 2 subtasks. In the first, we provide an overview of which phenotypes spatially interact with each other, and in the second, enabling the user to explore all the unique microenvironments. In the overview, for each tissue sample, we count the number of co-occurrences of each phenotype in its direct microenvironment and aggregate these counts over all cells of the same phenotype. Afterwards, we normalize it according to the amount of interactions per phenotypes of interest. To illustrate it, we use a heat map where each column represents the phenotypes of interest and each row the phenotypes existing in their microenvironment. On top of the heat map, we place the bar graph, showing the abundance of each phenotype. The more black a box of the heat map is, it represents cell phenotypes that interact more frequently. And these lead us to questions such as, which other phenotypes exist in the microenvironment of red cells when green cells are present? Do all the cells have homogeneous behavior? In order to answer such questions, we thought to map all the distinct microenvironments. Here we can see the example of 2 distinct red cells microenvironments. One has in its microenvironment blue and green cell, and the other yellow, green, and blue cells. In order to group all the cells with the same microenvironment, we use a modified version of the mathematical concept of motifs. We define that specific type of motif as a group of cells that share the same phenotype and they have in the microenvironment the same phenotypes. For the representation of a motif, we utilize a simple glyph based on a donut chart, with a circle in the center showing with its color the cell phenotype of interest. The colors of the arcs show the phenotypes that exist in the microenvironment and their size, the frequency that they exist in the microenvironment. To use it for visualizing the detected motifs, we need to extend the design to group all the microenvironments that belong to the same motif. In order to map the variation of the different microenvironments, we cut out part of the arc's color and we adjust the size of the inner circle according to the number of cells. Finally, to assist the exploration of the motifs in a small multiple grid view, we add a triangle to the top right of the glyph to indicate the significance of that motif. We enable the user to filter them using the overview heatmap. Selecting a box of the heatmap, it immediately filters the motif, retaining only those with a corresponding interaction. The expert can also further navigate among them, reordering them according to their significance, or the amount of cells grouped under its motif. All the aforementioned tasks have been implemented in an integrated system, with multiple linked views. For the remainder of the talk, I will give you a brief demo of our tool, based on a case study that can be found in the paper. We explore the spatial interactions among cancer cells and infiltrating cancer cells. To do so, we first normalize the heatmap per column. Then, we select cluster interactions via the boxes in the heatmap. This highlights the relevant areas and allows us to filter the resulting motifs. We adjust the range of the color map to reveal more detail in the heatmap. However, the patterns in the heatmap are inconclusive. Hence, we move on to the motif exploration. We set the threshold to the minimum number of cells that should be grouped in a motif. Sorting the motifs according to the frequency of occurrence allows us to locate cells that occur with high frequency and have only cancer and infiltrated cancer cells in their microenvironment. The two selected motifs meet the target criteria. However, the light blue cluster consists of significantly fewer cells than the orange cluster. Finally, clicking on the motif, we can relate it to its spatial location. Concluding, we have presented the workflow to enable the user in an interactive and data-driven way to precisely identify the cell phenotypes, explore and locate their spatial interactions, and identify subsets of phenotypes with significant spatial interactions.